Grand Rounds. It's my pleasure to have this morning with us Dr. Harish Vasudevan, who's an assistant professor in the Department of Radiation Oncology and Neurological Surgery here at UCSF. Dr. Vasudevan is a physician scientist who specializes in treating nervous system tumors in both children and adults. His lab investigates how normal growth signals, particularly in the RAS pathway, go wrong to drive cancer. Today, he'll discuss how his team uses cutting-edge functional genomics to identify new drug targets and design new therapies for patients with neurofibromatosis who are likely to develop tumors. It's a pleasure to welcome Dr. Vasudevan, and please welcome him to Grand Rounds. Thank you.
I should add, I did not write that intro. I don't think I would ever describe my own work as cutting edge. That feels like a bridge too far. But I appreciate the invitation and the opportunity to speak to everyone. So my name is Harish Vasudevan, physician scientist by clinical practice. I'm a radiation oncologist, actually usually on Thursday. So I moved my clinic this week to come over to Mission Bay. The rest of my time is spent running a lab where we are broadly interested in cancer genetics and understanding what I consider this duality of oncogenes and tumor suppressors, where many of them are cancer causing in the adult population, but also cause a number of developmental syndromes when they're mutated during embryogenesis. And one of the classic examples of that in my mind is neurofibromatosis, and that's been a big area of research for two main reasons. One, we have a fairly good biochemical handle on what the NF1 gene does, which is the causative mutation in neurofibromatosis type 1. And two, we have a lot of really great therapeutic approaches to take advantage of that biochemical handle. And so that has been a big part of what we've been doing. In that abridged bio, I'm actually not a Californian or a San Franciscan. I spent a lot of time in New York City before I moved out here. And I still maintain my New Yorker subscription, and this remains one of my favorite New Yorker cartoons. It actually came out back when I was still a medical student in New York, in the early days of machine learning, which has now been infinitely surpassed by AI. But I think the point of the cartoon is still super relevant, which is we have so much information. And I feel like every single day when I walk into work, especially in San Francisco, I'm attacked by billboards that are essentially telling me AI is going to solve all our problems. But I still think we're responsible for taking all that information and making it something that's useful for human knowledge and patient care. And so what I'll share today are a few stories of how we're trying to do that in this space. These are my disclosures. I will not be talking about anything that is relevant to my work with these different entities. Many of them are from years ago in grad school, actually. So I would like to start out by shouting out the people that make all the work possible in the lab and really give a high-level view of what we're trying to accomplish. Again, we're broadly interested in really understanding biologic mechanism of neurofibromatosis to build better diagnostics and therapeutics. This is a couple of pictures of the people on the team and the list of the folks in the lab. The one thing I always highlight is everyone in italics is a physician scientist, which means they cannot see my slides. All right. Yeah, please.
Thank you very much. I'm so sorry.
No, that's okay. Interrupt. Other people are welcome to interrupt too, just to be clear. Don't need to talk nonstop. See it.
Sorry. Yeah, sorry. No. Are you able to see him? Oh, no. Now? No, that's so weird. Give me one second.
Sure.
Go ahead. You can proceed.
Okay, thank you for the troubleshoot
I'm
that.
so sorry.
Despite how much technology we use in radiation oncology, I am often stumped by this type of stuff. So I'm glad we had someone to help. Yeah, I guess I'll just say feels bad to not highlight the people doing the work on the Zoom, but these are the people that do the work. We have a number of different folks in the lab. Everyone in italics is a physician scientist in some way, shape, or form. So they have a clinical practice. Devin's a radiology resident. Grant is a sarcoma soft tissue pathologist. Kanish is a neuropathologist, and Brian is one of our neuro-oncologists. So I think we really benefit from having the ability to go from the bench to the bedside and back. This is kind of our home base in terms of the biology we study. We're really interested in this fundamental catalytic interaction between RAS-GAP proteins, which are on the left side of this screen. NF1 is probably the founding member in my mind, causes neurofibromatosis type 1, which occurs in about 1 in 2,000 at least live births, one of the most common cancer predisposition syndromes. And coming back to that duality, about 20% of all human cancer has a mutation in NF1. So I think you learn a lot from understanding the development and the pediatric condition for how it impacts all adult cancers. RASA1 and RASA2 are similar members of the family. They have their own set of developmental syndromes. All of them have this shared catalytic or inhibitory function to turn off the RAS proteins, which probably need no introduction in this room. The most common oncogene in all of cancer. And one of the big questions here is where is the selectivity built in and how does this happen during development versus in disease? And for folks that are interested in RASopathies, thank you. That occurs in about 1 in 1,000 live births. So kind of if you lump all these things together, they're among the most common developmental syndromes folks are born with. And what do we actually do in the lab? Oh, I am lagged one behind on this screen. All right. We basically have 3 different sets of tools we'll apply, and I'll share some stories over the course of the next 45 minutes where we use these different hammers to find different nails. A lot of our studies start with patient samples here on the far left. Usually we're privileged at UCSF, especially in the brain tumor world, to have one of the premier biobanks. And so we're able to do a lot of work taking human specimens, generating testable hypotheses, and then going to our model systems, either vertebrate model systems, mostly mice, or in cell lines using CRISPR, which has made all of our gene editing so much more straightforward. I did my PhD in the pre-CRISPR era, and it took me a year to make some of the genetic edits I was trying to make. And now a lot of folks straight out of college make it in a month. And it's very, very, it's great for them. Maybe a useless skill set for me now. And the last thing, my postdoc mentor here is and was Frank McCormick. And so learned a lot about biochemistry and how we think about different proteins and use those insights to come with better treatments. And so a lot of signal transduction in biochemistry. Taking all those tools, most projects we can put into one of 3 neat buckets in the lab. The first is very basic neurofibromatosis and RAS-GAP biology. Often these are questions like which nucleotide or which amino acid on the protein mediates interactions with other proteins, and can we intervene on that? We're really interested in nervous system tumors. These are the most common tumors that arise in these patients. I put peripheral in parentheses because a lot of these things like malignant peripheral nerve sheath tumors are the most common cause of death in these folks. And kind of one overarching theme is we're always trying to look forward to develop better genetic and genomic technologies. One of my favorite types of projects in the lab is when we take a really old question and take a really classic finding and look at it through the lens of a new technology. And often we learn that scientists from 40 years ago were really, really smart when they came to their conclusions, but maybe we can add a little bit of mechanism and a little bit more context to that picture. So I did want to give a little bit of background on NF1, appreciating that this is pediatric grand rounds and many of you already know most of this. Progress in the NF1 space and in all clinical medicine, as I think all of you know, has a bit of inertia, but it has moved forward. And so in 2021, there are now revised diagnostic criteria for NF1, building on the 1988 initial NIH conference statement. Panel A and B here isn't necessarily to memorize. I actually have this printed up in my office on the side just to remind myself every time because it does change. What I'll point out is it's still predominantly a clinical diagnosis. So you need to meet a number of these clinical diagnostic criteria, 2 or more. It is one of those diagnostic criteria is having a NF1 heterozygous germline mutation. And so that is part of the diagnostic criteria, but if you read these diagnostic criteria strictly, that is not sufficient. So if you have just a germline mutation and no phenotype that is manifested, that is a gray zone, but by the definition, not the diagnosis. And then certainly we also have the hereditary component. The textbook answer is about 50% of NF1 is de novo, 50% is inherited. And kind of figuring out when that happens, why that happens, how it's different is something that we do think a lot about on the clinical side. As I mentioned at the beginning of the talk, NF1 is associated with a number of different tumor risks. Many of these are nervous system tumors like gliomas, malignant peripheral nerve sheath tumors, or neural crest-derived tumors, things like GISTs, pheochromocytoma, and then hematologic malignancies as well, JMML maybe being the famous example where a lot of great work has been done here at UCSF. And so this is kind of the range of phenotypes you'll see in these folks. And another big question we think a lot about in the lab is why do we have this large phenotypic diversity? I have folks I've seen with NF1 who are in their 80s and have lived a completely normal life and essentially have 7 capillary macules and 3 cutaneous neurofibromas and some axillary freckling. And then I have folks that have optic gliomas before they're 10 that I'm sure many of you have taken care of in the clinic. So we're trying to really understand, is there a way to predict that in the long term? And that's a big part of our work. And then I should have a shout out. I see Alyssa in row 2, so I'm glad I put the slide in here. We do have this comprehensive NF center that we have been slowly but surely growing over the past years. The clinical care component is on the adult side, Brian now, one of our neuro-oncology hires, and on the pediatric side, Alyssa, who has her clinic here one Thursday a month. Brian also kind of coordinates the education and outreach component of things. I run most of the translational research. And then we have many different folks in terms of a multidisciplinary clinic. And this is just a resource for folks to be able to click on after the fact if you need to refer someone or see someone that you're concerned about. So that's kind of the clinical background. Now, I am a physician scientist, and I often joke it's with a capital S. So now we'll jump to the science part of the talk. If there's one thing I can ask folks that are not basic science inclined to remember, it would just be this cartoon. NF1 is a RAS gap, which means it turns off RAS signaling. That is the only validated catalytic enzymatic function of NF1. If you ask ChatGPT or Claude or Copilot what NF1 does, you get a lot of other things. This is not the first thing it tells you. But there are 40 years of research that this is the only conserved biochemical catalytic reaction. So when you think about NF1 in your head, hopefully something about RAS goes off as well, and those should come as a pair. What does RAS do? It activates many things, among them RAF, MEK, and ERK, which can drive a number of cellular outputs. This is horribly simplified. It is a 6-week class to really go through this in any amount of detail, but suffice to say, this is the core RAS pathway in terms of how we think NF1 loss drives cancer. So you lose NF1, you get more RAS GTP loading because you don't have hydrolysis of the phosphate. You get more RAF, MEK, ERK, and all these syndromes I told you about. Whenever there are breaks, there's a gas pedal. So what's the gas pedal for RAS? There are a family of proteins called G protein exchange factors. They do the opposite of a gap like NF1. They take inactive GDP bound RAS and convert it into its active GTP bound form. So activating mutations in these gaps can cause cancer. There are now a number of SOS1 specific inhibitors that actually can be used for folks with SOS1 oncogenic variants. So there is both sides to this coin. And then maybe the most famous targeted therapies out there are actually against the very top part of this cascade, receptor tyrosine kinases. Tyrosine kinase inhibitors were in the textbooks when I was in medical school. And so they've definitely come a long way. And on that note, because of the fact that about half of all human cancers have a mutation somewhere in this cascade, We do have lots of great targeted therapies. Tyrosine kinase inhibitors, Gleevec was on the cover of Time. I always use that anecdote as something that was formative for me to see that a targeted therapy could change care and achieve that kind of notoriety. I don't think anyone gets magazines anymore, but the equivalent would be if Google decided to feature Gleevec on its homepage. I think that would be pretty motivating to a lot of people. RAS inhibitors are something that's relatively new. I used to list them, and now there are just too many to list, which is really a great problem to have. A large part of this work came from UCSF, from a number of groups here, so I think it's worth being aware of them. Many of these are RAS inhibitors that are specific for particular oncogenic variants, so it's not hitting wild-type RAS, they're hitting specific mutant variants of RAS, but that has been a huge shift in how we think about this. And then maybe some of the more common things we think of the NF1 world, MEK inhibitors, things like trametinib and selumetinib. There are certainly a number of RAF inhibitors, dabrafenib, often used for BRAF V600E mutant tumors. So we have a lot of different tools in this armament. That was all kind of the basic biochemistry. What about the genetics, specifically the human genetics? If I take everyone at UCSF that's been profiled by UCSF 500s over the past decades, 16% of all patients have a mutation in NF1. So that's, again, about 1 in 6 patients at UCSF. I'm a 20% clinician, and just by chance, I would see 6 patients on my random week, and I would then by chance see someone with an NF1 mutation. So I think if you see patients, this is a problem you have to deal with. The bar graph here shows the different kinds of tumors that arise in people with NF1. So the y-axis here is just the percentage of cases that have the NF1 mutation. The x-axis here represents the different tumor types. I started out by saying nervous system tumors are our focus, and those are certainly enriched with nerve sheath tumors and gliomas. Melanoma, a classic neural crest-derived tumor, same neuroectodermal lineage. But you can see that there's many other types of tumors that harbor NF1 mutations. So I think this ends up, again, being a cancer type agnostic problem that we have to deal with, kind of like RAS, right? RAS is mutated in many different cancer types, and the drugs that initially pioneered the way in lung cancer are now being expanded to other spaces. So I think there's a lot of value in thinking about it holistically. So I want to start out by talking a little bit about nerve sheath tumors, and we'll now kind of transition more to the research and data side of things. This is from a paper we wrote now 5 years ago, where we talked a lot about the different developmental lineages that give rise to these tumors. As I mentioned, people with NF1, we think, have a predilection for neuroectodermal derived tumors. So certainly the nervous system, but also the neural crest. The Schwann cell lineage arises from the neural crest. What we do know is that if you lose NF1 in the Schwann cell lineage, you get an entity called a neurofibroma. It's the namesake lesion for neurofibromatosis type 1. Typically, these mutations have biallelic inactivation of NF1, but have not yet accumulated anything else. There's a historical precedent that there's also a related syndrome called NF2 that I won't talk about too much today, but loss of the NF2 tumor suppressor causes a different Schwann lineage-derived tumor called a schwannoma. Kind of the nomenclature for these, if you go back and look clinically in the papers, is that initially, from a clinical perspective, there was a lot of challenge in actually separating out who had NF1, who had NF2. And so actually, Vic Riccardi, one of the guys who wrote that paper when he was at Harvard back in the 70s, told me once he also had an NF3 in his original paper. And it turns out that is actually Legius syndrome, which is another spread one related syndrome. So the clinical genetic folks really set the foundation for how we thought about this entity. Neurofibromatosis schwannomas, very different pathophysiology, but they do have a clinical behavior in common, which is that they are benign and they do not metastasize. Certainly can cause local compressive symptoms, certainly a big source of morbidity, but often not a cause of mortality. Kind of the dreaded complication is when these do transform into their malignant cousins or second cousins or aunt once removed, which is the malignant peripheral nerve sheath tumor. These are malignant. They do metastasize. They're the most common cause of death in people with NF1. The textbook answer is that about 10% of patients with NF1 will develop an MPNST at some point in their life. I can share that at UCSF, probably as a function of being at UCSF, that number in our data is about 30%. It's probably a selection bias on the kinds of folks that end up seeking care here. So if it feels like you're seeing more than 10% MPNSTs, I know Amit and I feel that way all the time. It's true. It's because it does come up. I'm a radiation oncologist, so I think for me, an image is worth 10,000 words. What I have here on the left is a classic example of a schwannoma. This is an axial T1 post-contrast image, and I hope everyone can appreciate the homogeneous to contrast enhancing ice cream cones arising from the vestibulocochlear nerve. This is essentially pathognomonic for NF2 schwannomatosis. In the middle in blue, we have a patient with a cervical plexus neurofibroma, well circumscribed, that kind of classic plexiform appearance. And you can see that it certainly doesn't look benign in a traditional sense, but in general, our clinical management of these is surgical resection if they're symptomatic, MEK inhibitor if they're symptomatic, and otherwise essentially watch and surveil and be thoughtful about when we're intervening. The dreaded complication is, again, this can transform to MPNST. This is actually the same patient 18 months later. And you can see that the tumor is no longer well-defined, doesn't have that plexiform appearance, and this came back with all the pathognomonic hallmarks of a malignant tumor. And this patient actually passed away within 6 months after that. And so this was actually pretty formative for me because I saw this patient, I think in 2018 when I was here as a PGY3, I guess. And that was something that kind of stuck with me and made me think that this was a problem we could intervene on. Because I remember doing the literature search for it and being like, there must be some way to predict. And then there was no way to predict at that time. So certainly something that we're still struggling with. Then, as I mentioned, there are currently no effective systemic therapies for MPNSTs or for recurrent neurofibromas or schwannomas. We do have some things we offer in the upfront setting that have efficacy, but if they grow through that or become resistant, don't have many options. And so that kind of set the stage for one of the papers I wrote during my postdoc, which is we took 119 peripheral nervous system tumors. This is a mix of schwannomas, neurofibromas, and MPNSTs. And we performed methylation array profiling. Why methylation arrays? There's two reasons. One, from a basic science perspective, methylation to me is a reflection of cell lineage. So you get a really, really robust readout of where these cells might be arising from. The second thing is it's part of the classification system for central nervous system tumors. So the WHO 2021 update actually put this in as part of the classification system. So it stood to reason that it would also be useful for peripheral nervous system tumors. And we'll come full circle at the end of the talk about how we're actually trying to apply this. In our initial study, we identified 3 groups of tumors. I'll go in reverse order. I'm very bad with naming the groups. I call them group 1, 2, and 3. And the last thing the reviewer asked me to do was rename the groups. So I renamed them and then I put them back as 1, 2, and 3 after I put the actual paper in, just to like make sure that we didn't have any confusion around it. Group 3 are essentially all schwannomas. You can see the histology in that second row there is that light green. These are almost all enriched for NF2 loss, which is that bottom row there in the mutational plot of this oncoplot. Group 1 and group 2 are where things are a little bit more interesting. These are all MPNSTs, but there are different methylation subgroups of MPNSTs. And at the time, we didn't appreciate this, but now that we've started to have more patients and more samples, we actually think this has to do with syndromic versus sporadic tumors. So again, I'll touch on that at the very end of the talk. Malignant peripheral nerve sheath tumors, the textbook answer again, 50-50. If you're in the NF1 world, that's usually a good guess. 50% sporadic, 50% NF1 associated. Historically clumped together, probably there's a molecular difference based on their pathogenesis. So this is all really great. We have a classification system. Turned out these methylation groups were not prognostic in our cohort, and we have struggled to find a prognostic methylation biomarker today. So this is probably not the solution for prognosis. But the question we had was, why were we seeing NF2 in all of these different tumor types, even if you look all the way on the right at the neurofibromas and MPNSTs? And maybe more directly, we know MEK inhibition, which is now FDA approved for neurofibromas, is helpful, but there are a subset of patients that are either intrinsically resistant or stop responding. And our goal was really to understand the mechanism underlying that resistance. And so I'll just show one slide because the work is now published. And for folks that are interested, I'm happy to talk about it afterward, or you're welcome to read the paper. What we did was we did a genome-wide CRISPR interference screen to look for mediators of MEK inhibitor response, in this case, selumetinib, which at that time was the only FDA-approved MEK inhibitor in neurofibroma cells. So the way we set up this experiment is we have the dCas9 crowd BFP. This is a dead Cas9. So essentially, depending on what small guide RNA you have, it goes and sits on a specific target gene and prevents the gene from being turned on. So this is a very, very efficient way to screen genome-wide all of the different potential genetic perturbations. We then transduce in a genome-wide small guide RNA library. So each cell on average gets one guide knocking down a specific gene. And then we treat them either with drugs, selumetinib, or vehicle. And then we can ask what types of genetic perturbations either promote resistance to selumetinib, so there's more of those cells at the end, or sensitivity, there's less of those cells at the end. This is one way to look at that data. So the x-axis here is the ratio of sgRNA counts in the drug versus control condition. The y-axis for log 10 of the p-value, classic volcano plot. Anything in red on this plot on the right, knockdown or repression promotes resistance because we saw more guides with it. Anything on the left in blue, knockdown promotes sensitivity. And one of the things that jumped out from our human data as well as in our CRISPR screen was this recurrent alteration NF2. And I'll leave the story there, and that if we remove NF2, that is indeed sufficient for MEK inhibitor resistance in this context. And in the paper, we speculate a little bit more about how this is happening, why this is happening. There's kind of crumbs of this throughout the literature. But I do think this is one mechanism of MAP kinase independent resistant to MEK inhibitor with another tumor suppressor that actually happens in people. So I think there's value to this. So that's kind of where we were when I started the lab now 2.5 years ago. And so what I wanted to share are 3 stories that are different phases of getting out into the world from the lab. So the first stories, we'll be talking about glioma and glioblastoma, thinking about prognostic and combination therapeutic strategies. The second story, which kind of scratches the biochemical itch I always have, is can we do better than MEK inhibition? I showed you on that initial graph that NF1 turns off RAS, and there's this cascade to RAF and MEK, but it stands to reason they could go right to the source and hit RAS directly. And then the third story I want to tell is this evolutionary process from a neurofibroma to an MPNST, and how can we really understand that on a cellular and a molecular level using spatial transcriptomics, which has been all the rage for the past 5 years. And I think we're just publishing as that wave starts to crash. So hopefully we'll take advantage of it. So I want to start out with the glioblastoma story. And when I was thinking about whether I needed background, I thought it was, I've never been told I have too much background, only too little. So I figured I would add a little bit extra background into the talk. Glioblastomas are the most common malignant brain tumor. This is from the CB Trust, which is the Congressional Brain Tumor Registry that's run through the United States ever so often. We have a joke that Quinn Ostrom is great at publishing a 25,000 plus citation paper every 4 years, right? Because this gets cited by every single person that needs to talk about brain tumors. The malignant tumors are in red. And if you look up there, glioblastoma makes up almost 15% of all malignant brain tumors. And UCSF, certainly our neurosurgery department, has a really long history of being aggressive with these tumors, pioneering a lot of the surgical methods. And so we just have a very unique biorepository of these samples. NF1 has many, many claims to fame, but I think one of them, it was actually the first gene identified in the Cancer Genome Atlas. So this is a cartoon from the initial Cancer Genome Atlas paper published in Nature back in 2008. And the first tumor they tried to identify was actually glioma and glioblastoma. And the first gene that came out of that, if you look kind of at the conference proceedings back then, is NF1. So NF1 is mutated in 18% of all glioblastomas. Despite that fact, we do not have a targeted therapy for that alteration. We do not treat those patients any differently. And so kind of when we started out this project, that was where we came from. And then I'll just say I kind of move always back and forth from the developmental germline NF1 syndromic condition and the somatic NF1 adult tumor condition. All of the data I just showed you were somatic adult tumors. This is a paper that CJ Lucas, one of our former neuropathology fellows, now a neuropathologist at Hopkins, put together a few years ago, understanding syndromic NF1 mutant tumors. So these are gliomas arising in people born with a germline mutation in NF1. The way I think about this is very similar to how I think about the Schwann cell neurofibroma MPNST transition. You have presumably a glial lineage cell, although even that is up for debate these days, where it's NF1 heterozygous. You lose the second copy of NF1, you get a low-grade glioma. It might look something like this on this T1 postcon, in this case in the posterior fossa. You can appreciate this heterogeneously contrast-enhancing mass on the right. Additional genetic alterations, in this case, CDKN2AB and ATRX, after the initial surgery, the tumor came back as a glioblastoma. A big part of this paper was trying to understand, are there molecular subgroups of NF1-associated gliomas? I wouldn't be telling you if the answer wasn't yes, we think there are. And so in this particular study, CJ was able to identify a high-grade and a low-grade group, the high-grade group in yellow showing significantly worse glioma-specific survival. And so this is now part of our clinical practice in terms of how we think about these tumors. The reality is when these tumors are low grade, they're often stable and we're not taking them out. And the times we take them out are when they're high grade and aggressive. So we're probably enriched for high grade tumors in this type of a study. But nevertheless, it gives us a little bit of guidance on postoperative therapy like radiation. And so what did we want to do? We wanted to take advantage of the fact that we have way more somatic tumors than syndromic tumors. That's just a fact of the matter of how the epidemiology of these studies work. So Charlene, grad student in the lab and Canadian neuropathologist in the lab, paper actually just came out earlier this month in JCI Insight for folks that want to read about it. Took 186 NF1 mutant IDH wild type glioblastomas, which are grade 4 gliomas, and looked at the mutational spectrum. I put the oncoplot here just to show how much work it was to get this oncoplot together, but not very useful for a talk. Another way to think about this is just by looking at a bar graph. So what we have here on the x-axis are all the different co-mutated genes. The y-axis is the percentage of cases that have that co-mutation. And red is our NF1 mutant cohort, and blue is the NF1 wild type propensity score matched cohort. And there's two things that pop out. One is, in general, not a lot of differences in the co-mutated genes. One hypothesis might have been you need certain types of co-mutations when you have an NF1 mutation versus not. That does not seem to be true. The second thing is there is this mutual exclusivity between NF1 mutant tumors and receptor tyrosine kinase mutated tumors, EGFR being the biggest offender there in blue. This has been previously reported, and the hypothesis has been that when you have an RTK mutation, that dominates over the NF1 mutation. Another thing that a few people have speculated about in the literature is that NF1 mutant tumors may actually be badder actors or worse actors than NF1 wild type tumors. If you look at the original cancer cell papers from the TCGA, there's some data suggesting that. In our data, the survival curves crossed many times. I don't think I've ever had a p-value closer to 1. And so I think there was no difference between these two big picture subgroups. One thing we did see, though, was a difference in the effect of CDKN2AB loss. CDKN2AB loss is very interesting from an NF perspective because it's thought to be the second hit that takes benign tumors in people born with NF1 and puts them down a malignant trajectory. We actually see 9p loss. We'll come back to this at the end as well as one of the earliest indications of malignant transformation. We even made a name for it, atypical neurofibroma of unknown biologic prognosis. It's a mouthful. We need to change it. Told you I wasn't good with names, but CDKN2AB is that second hit. What we saw was in NF1 mutant glioblastomas in panel D here on the left, there was a significantly worse outcome if you had a CDKN2AB co-deletion compared to CDKN2AB intact tumors. That wasn't super surprising. CDKN2AB loss is known to be a bad molecular prognostic factor in gliomas. What was surprising is that NF1 wild type tumors did not have that same trend. The reviewers really did not like this, and so they made us look at a bunch of other data sets, and I'll just show one of them, which is we see the exact same thing in the Cancer Genome Atlas data. Why is this at odds with what maybe other people have shown? I think there's two explanations. One is we restricted our analysis to IDH wild type tumors. It's not a new molecular biomarker, but it's new compared to when the Cancer Genome Atlas studies were originally done. So I think we have a different subset. The second thing is the criteria for NF1 mutation. NF1 is a really, really big gene. It's about 8.5 kb in cDNA length for folks that think about cloning things. I saw a couple of people with big eyes when I said that, and I think that says it all. So I think there's a lot of passenger variants. We were very stringent in how we filtered those versus earlier studies were not. So maybe two possible explanations for why this was not observed earlier. So that's kind of the clinical observation. But as I mentioned before, we're always trying to take this back to the bench and understand how this guides therapeutic development. And so similar to what we did in our neurofibroma world, we set up a panel of genome-wide CRISPR-I screens. Again, exact same setup. We were able to screen every single gene in the genome and assess response to MEK inhibition, in this case, selumetinib. We took two different cell lines, a mouse and RAS mutant cell line. It is an oversimplification, but in general, we think of RAS activation in these contexts as copying NF1 loss. It's probably not entirely true, but for the purpose of this study, we started with that. We also had a human patient-derived NF1 mutant glioblastoma model. So we did the screen in both cell lines, and I'm showing the data slightly differently here to look at the overlap between these two screens. So what I have on the x-axis are all of our hits for the mouse SB28 glioblastoma line. On the y-axis, all the hits for the human glioblastoma 43 cell line. All the dots in red in the top right quadrant are conserved resistance hits. All the dots in blue in the bottom left quadrant are conserved sensitizing hits. You'll probably notice two things: much fewer sensitizing hits and resistance hits. And overall, not a whole lot of concordance between the two cell lines. I think this is a feature of every single screen we've done, but we really want to focus on the conserved biology. And so what were these shared hits? The two top ones were BRAF, which probably needs no introduction to this group, direct downstream target of RAS. The other one that was interesting to us was a protein called SHOC2. And so I have a schematic here of the RAS pathway. RAS activates BRAF, and we think that SHOC2 is a critical part of a complex that regulates BRAF. So essentially, the way you sensitize these cells to MEK inhibition is by turning off the pathway more. And so while we were doing this, it turns out we weren't the only people interested in SHOC2. There were a lot of people interested in SHOC2. So there were a trio of papers in Nature that came out as we were kind of in the middle of this story, all of which solved the structure of SHOC2 and really nailed it down as part of this heterotrimeric complex to dephosphorylate and activate the RAF protein. So again, this is at least my home base. NF1 turns off RAS, RAS activates RAF, MEK, and ERK. SHOC2 functions as a complex with mRAS, so not one of the classic oncogenic RAS proteins, but another one that has a RAF binding domain and can bind RAF. And in this example, it's the PP1 catalytic subunit. In reality, any phosphatase catalytic subunit can swap in for this. And what this complex does is remove an inhibitory phosphosite off of RAF. And so when this complex is present, RAF can signal because this phosphosite has been dephosphorylated. When SHOC2 is gone, you no longer have that dephosphorylation. Now RAF is stuck in an auto-inhibited conformational state. And these three papers, there were hints of this until these three papers came out, but these kind of really nailed down the structural basis for this, and that this indeed was the mechanism. So we didn't get to do biochemistry in this study because they kind of did all the biochemistry before we could get to it. So what we decided to do was pivot and look at therapeutics. And so as a simple proof of concept, we CRISPR'd out SHOC2 in our cells and looked at the response to MEK inhibition. What I'm showing here are two sets of immunoblots with a dose response to the MEK inhibitor selumetinib. The top row is SHOC2. You can see our SHOC2 knockout cells don't have it, as expected. The second row, which is maybe the one to really pay attention to, is phospho-ERK. We use this as a readout for sensitivity to the MEK inhibitor. And what you can appreciate is when you're SHOC2 deficient on the right, you have about a 1.5 order of magnitude less sensitivity to MEK inhibition. One of my PhD advisor's favorite statements was show it in at least 3 different models. Here's model number 2, where we see the exact same thing. And then our third model is actually in vivo. So what we were able to do with these mouse SP28 cells is actually implant them intracranially in an immunocompetent host mouse and assess the response to MEK inhibitor. The way we do this experiment is at day 0, we implant these SP28 glioblastoma cells intracranially at the skull base. We let them establish for about 5 days, and then we treat, in this case, with the MEK inhibitor selumetinib until 21 days, which is our experimental endpoint. And so what does this look like? The x-axis here is time, y-axis is survival, classic Kaplan-Meier analysis. The black line here is the control cells. They just got a scramble CRISPR-I sequence. Those mice are all dead by about day 15. The gray is just the effect of the MEK inhibitor selumetinib. You see a little bit of prolongation. The dark red is the effect of SHOC2, a little bit longer, and then really the combination, you get them out to 30 days, which in this assay doesn't seem like much, but in this assay is a big deal. And I'll remind everyone, median overall survival for glioblastoma, even in 2025, is on the order of a year. So these aren't people that are living a very long time. So I think any survival benefit is very meaningful. So hopefully I've convinced you MEK inhibitors at least have some efficacy, and there's some ways to improve on the combination, but you're also all probably wondering why doesn't it result in a stable response? And the answer for most targeted therapies is tumor heterogeneity. What we mean by that is a tumor is made up of millions to billions of cells, depending on how big it is. No two of them are quite the same genetically, epigenetically, transcriptomically, or proteomically. And people have done a lot of great work in the glioma world defining what those cellular subpopulations are. So this is the graphical abstract from Mario Suva's group published in Cell in 2019, where they defined 4 transcriptional subgroups of glioblastoma tumor cells. We have OPC-like cells in green, NPC-like cells in blue, AC-like cells in yellow, and MES-like cells in red. These are all developmentally defined. So coming back to this link between development and cancer, OPC is oligodendrocyte precursor cell-like, NPC is neural crest precursor cell-like, AC is astrocyte-like, and MES is mesenchymal catch-all. What they were able to do in the study really nicely was show that these 4 cell types exist in pretty much everyone's glioblastoma. So it's not like you're just made up of one of these cell types. And they were also able to show, using some really great informatics, that there are relationships between these transcriptional cell populations and DNA mutation. And in 2025, DNA mutation is still what we get on every patient. So we're not really getting this level of information. So I thought it was really nice to tie that together. The question we wanted to ask is which cell populations actually underlie resistance to selumetinib? You can envision one of two models. Every cell type is equally sensitive, and by hitting the MAP kinase pathway higher, you equally decrease the sensitivity or resistance of each population. The other model would be one cell type you deplete completely with the MEK inhibitor, and the other models don't give a lick, and they're the ones that come back. And so we set up an experiment to do this, which is we took that same mouse glioblastoma tumor model and treated them with selumetinib, but then single cell RNA sequencing at the end to essentially define these cell populations in our treated tumor model. For folks that do a lot of single cell sequencing, this is really for y'all. Each dot here is a different cell type, and the relative distance between them is how similar or different they are in gene expression space. The reason I draw this is I feel like people often skip this step and just show you the final plot. Most of the cells we got from these mice were not tumor cells. And we can do this because one of the clever things we did in the lab a few years ago, postdoc's idea was to make all our tumor cell lines male and make all the host mice female, because you can house them together, and then you can just separate based on exist expression, which cells are from the tumor and which cells are from the host. And so, you know, that avoids all these conversations people get into about how do you know what is a tumor cell. I think that's more philosophical than informatic, but with this approach, we can just say these are the tumor cells. And so for all the rest of the analysis, we just restricted it to these tumor cells because the microenvironment, I think, is a different beast that we won't talk about today. So when we restrict it to that analysis, we can essentially take each of those tumor cells and give it a score in this 4-quadrant space to assign it one of those single cell states that Mario Suva's group initially published. So you have the mesenchymal-like cells here on the bottom right, consistent with what Mario's group saw. The NF1 mutation does enrich you for mesenchymal-like cells, about 82%. And then you have a minority of these other cells. And one of the first things we asked is there a difference between these NF1-associated mesenchymal-like cells and the rest of the cell types in terms of their sensitivity to MEK inhibitor response? To do that, we informatically constructed, using our CRISPR-I screens, a signature that predicted sensitivity to MEK inhibitor selumetinib. And what you can see is that MES mesenchymal-like cells are significantly more sensitive than non-mesenchymal-like cells. So kind of model 2 for folks that were paying attention to the 2 hypotheses, right? Different cell types have different sensitivities. And this is just to say, what does that really look like? And so what we can also do is look at gene expression changes following MEK inhibitor in the mesenchymal compartment. And the mechanism of resistance in that compartment is upregulation of the MAP kinase pathway. You see things like NRAS, FOS, Jun, DUSP, classic RAS transcriptional targets. So for the mesenchymal cells, they're really addicted to RAS signaling. So when you hit the pathway harder, you're going to deplete that population. Not quite what we see for the non-mesenchymal-like cells. So here we actually see upregulation of what I would speculate are dedifferentiation genes that are driving you backwards in glial space. So things like SOX10, PGF alpha, the OLIG genes, PTPRZ1, which is a well-established glioblastoma stem cell marker. So you have 2 different resistance mechanisms, one of which would probably benefit from hitting the pathway harder, but one of which probably wouldn't. And so I think this is part of our toolkit, but probably not everything. And so based in part on our work and many other people's work, we've been slowly, as Alyssa can attest, moving this clinical trial concept forward to look at combining MEK inhibition with radiation. One good thing about radiation is, in general, it doesn't care about the genotype of your cell. There's some exceptions to that, but for this context, I think it kills everything that's dividing equally well, damages the DNA just fine. So the idea was we can maybe combine MEK inhibitors which take advantage of the NF1 loss, but with radiation therapy. And so we've set up this trial basically with 3 cohorts. We just did the 4th line by line last Friday. So I'm learning what that process is like. And it's exactly what it sounds like for folks that haven't done it. You read every line with a group of 6 other people and make sure everything is clear. We have 3 cohorts in this trial concept. Cohort 1 are folks that are NF1 mutant, not germline, just somatic, that have previously gotten radiation. It's really hard to run a trial for newly diagnosed glioblastoma just because we know radiation and temozolomide is the best we have to offer, and it's really hard to maintain equipoise and offer something else. So almost all glioblastoma trials right now are in the recurrent setting. So this is for folks that are progressed after standard therapy, and the idea here is that we would combine radiation with MEK inhibitor. Cohort 2 are folks with germline NF1 that have a glioblastoma, so the highest grade tumor we can give them. And these folks we're also treating in the upfront setting with MEK inhibitor and radiation. The idea being that there is a track record of MEK inhibitor efficacy in these folks, and radiation can maybe deliver some of the knockout punch. And then the 3rd cohort, is not radiation related, which is perfectly fine by me. My favorite therapies for kids are targeted therapies too, even as a radiation oncologist. And so in this cohort, we're essentially treating folks with gliomas with MEK inhibitor only. And yeah, I think we're slowly getting there. We've found alternate sources of funding for it as the DoD consortium collapsed. So I think there's now money for it and interest in it. So I'm hoping Q1 2026 is our new update. So if you ever have folks with these mutations, we'd be happy to talk to them about it. So I will skip this slide in the interest of time and just jump to this. So the other two stories are much shorter, so do not be alarmed. We will be done by 1 p.m. The second story is thinking about how we can do better than MEK inhibition by directly targeting RAS. This is the fourth time I'm showing this cartoon just because I think it is so essential to all the work we do. NF1 turns off RAS. The textbook answer is RAS activates RAF. Again, if you ask ChatGPT, RAS can activate many, many other things as evidenced by this question mark on the right. Probably the best established one is PI3 kinase. There's actually been, I think, a half dozen papers in Science and Nature this year about perturbing that interaction between RAS and PI3 kinase using small molecules. So this is something that's in the clinic now. There's other things RAS can signal to, and the idea would be that if we inhibited RAS directly, we would kind of block all of it. In contrast to a MEK inhibitor, which is 2 steps away from the causative NF1 mutation and wouldn't take care of any of this other direct RAS proteins. The other elephant in the room I should mention is that NF1 itself also has a bunch of other reported roles. Again, AI can enlighten you. None of them are super well established, but I think it would be important to think about it from first principles to make sure there wasn't something else we're missing with a RAS inhibitor. So what do we do? I think one of the challenges with NF1 is its interaction with RAS is so transient. Essentially, once NF1 finds a GTP-bound RAS, that hydrolysis happens in pico to nanoseconds, and the whole thing falls apart. So there's actually never been a study that's been able to find NF1 and RAS in complex in a cell. People have tried really, really hard. A lot of really smart people have tried really, really hard using conventional APMS and a few other approaches. We tried to take a slightly different approach, which is a proximal proteomic approach. There's a couple of different methods out there, APEX being one, BioID being one of the other ones. For mainly familiarity reasons, we went with APEX. The way this type of an experiment works is you take your protein of interest, you fuse it to the APEX tag, and then you essentially treat with hydrogen peroxide. And so the APEX converts all the exogenous biotin phenol and essentially has a cloud of about 20 nanometers around it and captures everything that's in proximity. So you have spatial specificity. It's not just stuff that binds through some complicated complex. And then after 30 seconds, you're then able to actually pull down on the biotin and get everything that was in that cloud. So you have temporal specificity, 30 seconds. This is really, really valuable for things that are transient because now you really kind of set yourself up for success. So it took about a year of optimizing to get NF1 tagged in the right place. NF1 is big, it dimerizes, it has to get to the membrane. All of these things can be disrupted, but we eventually got it to work. And we set up a pretty simple experiment, which was we took peripheral nerve cells. So these are just wild type cells from a healthy donor who donated their peripheral nerve to science. We put in our APEX tagged NF1 construct, let them hang out for 24 hours. Treated them with a MEK inhibitor, selumetinib, for 30 minutes. The reason for that MEK inhibitor treatment is when you treat with a MEK inhibitor like selumetinib, you turn off the feedback. And so you get a lot more GTP-bound RAS building up in the cell. So you're setting yourself up again for success. Then we did this 30-second hydrogen peroxide treatment and saw what came out. This paper is actually not published too, so I kind of was good timing to give this talk because not people can read about it. But I'll just talk about the very end of the paper, which is we looked at all of the different hits. So anything in red on the right are things that were in closer proximity to NF1 after MEK inhibitor treatment. Anything in blue on the left are things that were depleted. And one thing that jumped out to me right away when I was looking at the Excel sheet at like midnight was that KRAS was at the very top of the list, and I immediately control F HRAS and NRAS, and they were somewhere else on the list. And so that's super interesting for 3 reasons. One, there's really no established mechanism of biochemical selectivity. The dogma is that all the RASs are needed for NF1. So this is a really nice example. Maybe that's not true. We have some still unpublished structural data that actually NF1 might make contacts with KRAS on the back end as a full-length protein. I think this was missed because most people don't work with full-length NF1. So if you look at just the catalytic domain of NF1, that can bind all the RASs. The third reason is pan-RAS inhibitors. There is one in clinical trial for pancreatic cancer that's probably going to go the distance, but really toxic. You can imagine blocking all the RASs is not great for humans or mice for that matter. So if there was a way to hit just one RAS, that could really open up the therapeutic window. There's pretty good mouse data that ablating KRAS inducibly in the adult is fine. Those mice tend to do with just, there's some small behavioral things from not having KRAS, but overall they live a normal life. So for those reasons, we were pretty pumped about this result. So the first thing we did was do the pharmacologic experiment. We took a KRAS inhibitor, treated NF1 mutant neurofibroma cells, these NF9511B line from a patient, and asked, is KRAS inhibition sufficient to turn off RAF, MEK, ERK signaling? The answer was yes. And then the other question we asked is, does it affect PI3 kinase AKT signaling? The answer seems to be no. We actually now think that PI3 kinase as a target of RAS may be mostly restricted to oncogenic contexts, not when you have wild type RAS.
Can you comment on what type of RAS inhibitor this is? Because a lot of them work by blocking the RAS-RAF interface, right? So you wouldn't expect there to be a PI3K effect.
Yeah, so this is it's the BI inhibitor. And so the way that inhibitor was designed is it's a Nature paper from a couple years ago. I'm happy to send over, but essentially they took all the residues that were unique to KRAS, not for HRAS and NRAS, and optimized a compound to bind those. It brings up a really good point. I use RAS inhibitor pretty loosely, which isn't great. There's G12C inhibitors, which are the ones that are in the clinic, the only ones that are in the clinic right now that are FDA approved. So those are only for patients with a G12C mutation. There's pan-RAS inhibitors which hit H, N, and KRAS. There are now, that one again is in the clinic in a trial for pancreatic cancer, which we think is wholly RAS driven. There are KRAS specific inhibitors, and then even within the wild type KRAS inhibitors, there's GTP specific inhibitors and GDP specific inhibitors, because there's context where you might want to hit just the active or the inactive conformation. And then there are a couple of now NRAS specific inhibitors that are in preclinical testing, which is for melanoma would be huge. So yeah, this was the BI's inhibitor, but I don't think it's, we've tested a few other ones and we see similar, but not the same results, which I think is because of the GTP-GDP issue. And then the last thing I'll say about this story is that we did PTM mass spec to confirm that KRAS inhibition is indeed sufficient in this cell line. So we took those NF1 mutant cells, treated them with the KRAS inhibitor for 30 minutes and did PTM mass spec, mainly to look at off-target effects, but also the effect on cell cycle. And what you can appreciate here, everything in blue on the left are kinases that go down when we treat with a RAS inhibitor. So you see ERK1, ERK2, like our blots. You see CDK1, CDK2, so it's sufficient to block cell cycle progression. And you actually also see PKC and PKA, which is an area of ongoing investigation, because if you look, again, I always joke about asking ChatGPT, but it really is the best way to get a quick answer. It'll draw a circle for you of how RAS and PKA regulate each other. So, all right, and then the very last 5 minutes here, I just want to talk a little bit about spatial transcriptomics and diagnostics. I feel like I harped so much on the fact that we can't tell which neurofibromas become MPNSTs. So we should probably be trying to do something to solve that problem. And so the way we think about this transformation process, this is a cartoon from a review that a bunch of us put together earlier this year, is you have neurofibromas, you have these atypical neurofibromas of unknown biologic prognosis, the anapy in the bottom left, and then you have an MPNST. And we think of this as a stereotypic sequential transformation process. In reality, it is parallel, polyclonal, and branching in every single which way. And so to try to bring some balance to that force, we've taken the histologic characterization, which is historically what we've done, and tried to overlay molecular analysis. And so the only thing we could all really agree on was that we should do molecular analysis. We agreed that if NF1 was the only mutation, that should keep you as a neurofibroma. We also mostly agree that if CDKN2AB, that second hit, was deleted, that should upgrade you, even if histopathologically you look like a neurofibroma. And then similarly, we also agreed SUS12E and P53 mutation should upgrade you to the malignant peripheral nerve sheath tumor. This significant aneuploidy term was added in kind of at the 11th hour and has created a lot of distress, because how do you define aneuploidy and how do you define significant? We didn't offer those definitions in the review. So we actually set a patient this morning at our NF tumor board where we were talking about, they were upstaged to MPNST by the NCI group solely on the basis of significant aneuploidy, but didn't have CDKN2AB, SUS12E, P53. And the patient basically had a surgery and was fine for the next 2 years. So probably that wasn't an MPNST. So I think there is still a lot to learn, but this is our framework right now. And for anyone that's a pathologist in the room, these are the examples of what histologically might look like and what the molecular features might look like for folks that get upstaged by the molecular data. And so what did we want to do? I think one of the big challenges in all cancer is the amount of heterogeneity there is in these tumors. You don't really ever know what you're biopsying, even when it's image guided. And even if the biopsy comes back as non-diagnostic or with a diagnosis, and you look at what's left behind as a radiation oncologist, I'll see the diffusion restricted part of the tumor left, or I'll see a PET avid part of the tumor left, and it's hard for me to tell exactly what got sampled. So I think we have to be really thoughtful about that. And one way to do that is to do something called spatial transcriptomics. Essentially what this lets us do is it lets us take an H&E slide and do gene expression profiling at every single spot on the slide to then connect histopathology to transcriptomics. We can also infer microenvironmental populations. We can also infer copy number changes at the chromosomal level. And so Kanish, who's a now neuropathology faculty member, which you'll probably see him around, set up this project where we took 15 MPNSTs, 11 were NF1 associated, 4 were sporadic, and we did spatial transcriptomics to understand what that distribution looked like. This is the oncoplot of all 15 patients. I'll just draw your attention to the boxes I've circled in red. We had 2 specific patients with NF1, N3, and N4, not super creative, who had transition zones, which we defined as an H&E slice where there was a part of the sample that met the histopathologic criteria for neurofibroma, and a part of the sample that met the histopathologic criteria for an MPNST, and then an intervening region. And so I think this gave us a really cool opportunity to apply this method to understand evolution happening in real time on a slice. So what did we find? This is what that data looks like to our pathologist on the left as an H&E. Kanish and a few of our other neuropathologists went on and annotated the areas that were low grade in light green. High grade is in the bottom right, and the transition zone is everything else. If we look at our gene expression profiling and look for gene expression clusters without telling it where things are in space, they actually cluster spatially, which makes a lot of sense for biology, right? Things that are close to each other should kind of look the same. We were able to identify 6 clusters in this example. And if we do something called pseudo time analysis, which lets us order the events, you actually see something interesting, which is a branch trajectory. So when you do this analysis, the 2 things you got to give it are the gene expression data, and you have to give it a time 0. We have the unique advantage in this case of knowing that the tumor had to start at the low grade. That's biology. So we gave it C2 as the time 0. And what I hope you can appreciate in the bottom right is that C2 bifurcates into 2 different trajectories, one to cluster 4 and one to cluster 5. And that actually correlates with the with the spatial transcriptomics and the H&E to malignant transformation. This is that second transition zone sample, very similar setup, low grade transition zone, high grade. We see spatially distributed clusters and we see bifurcation, in this case, after an initial evolutionary process into the atypical part. So that's all great, but like what is the actual biology of this? And so in the last minute, I'll just share, there's 3 things we see. First, as our pathologists have been able to tell us for 150 years, there is more mitotic activity in the areas of the tumor that are malignant. That's what I'm showing in the top left. You see a lot more G2M and S phase cells. You also see dedifferentiation. Again, I say it so many times, but I really think developmental cancer are just going backwards. So you see loss of S100B and SOX10, which are classic Schwann cell markers over time. So essentially the low grade area retains these and you lose them when you become malignant. You also have changes in the microenvironment, specifically in the CD68 macrophage population. We think of this as being at a medium level in the low grade, spiking in the transition zone, and then going away when you're in the malignant part of the tumor. And then the last thing is we can infer copy numbers. 9p is where CDKN2AB loss, that gene lives. And so you can see that it is intact in the low grade part in gray, and then it is polyclonally lost, and then clonally lost in the high grade part of the tumor from top left to bottom right. Why does this matter? I think CDKN2AB loss should be considered necessary, but not sufficient for transformation, because there's clearly cells that have it, but don't transform. And then same game here. So I think I'll go ahead and stop there and just summarize, add 1 slide, but I hate when other speakers go over time, so I will not do the same. Really, 4 things I tried to tell you. NF-associated tumors have a lot of heterogeneity. All of cancer has a lot of heterogeneity. The main thing here that gives us a unique ability to study it is we know what the first germline alteration had to be. So we have a common starting point. I'm a big fan of functional genomics. I did an RNAi screen back as a grad student, which is one of the most painful things I did. CRISPR screens have really opened up the door. Like we've done a dozen of these, and I'm happy for anyone that's basic science inclined to help set that up for you. Proteomics are really powerful, and we're now working on a trial concept to use KRAS inhibitors in this setting, because I think This could be really valuable for two reasons. One, maybe for patients that don't respond to MEK inhibitor, you have an alternate pathway. And two, we speculate the toxicity profile might be better because it's more selective. You have two other RASs as backups, versus most MEK inhibitors have both MEK1 and MEK2, so you don't really have a backup. And then we're working on taking that spatial transcriptomics data into a biomarker. The way we're doing this is actually I made fun of AI at the beginning, but I'll say I've embraced it. We're taking those spatial transcriptomic clusters and using AI to learn on the H&E section exactly what the H&E looks like for each of those groups, and essentially setting up a score for how likely we are to have these malignant events happening, and then validating that on an external set of H&Es where we don't have the spatial transcriptomics, but we have the clinical annotation and the power to really ask that question. So that's where that is going. So with that, happy to take questions. Anyone that's interested in hearing more, I'm always happy to talk about science and clinical questions that we could maybe help out with. So these are all the people that make the work possible, and thank you for your attention. Go for it.
I have a question about the potential use of patient tumoroids
Mhm.
in this to study these transition points
Mhm.
in dedifferentiation.
Yep.
Are those being used in your lab, and how are you kind of employing that idea?
Yeah, so the question was about patient tumoroids and organoids, I guess, to put them in that same kind of bucket, although I appreciate those are different things. We have tried. One major challenge is neurofibroma cells are benign and don't like to grow. And so it's actually non-trivial even to just set up monolayer cell lines with those. And so we have not made a ton of progress in 3D cultures or tumoroids from those. We do have some with MPNSTs, but that doesn't let us answer that question that you asked, which is really, really important. The place we have made some progress on this is actually the glioma part of the work. There's a lot of really nice systems with astrocyte glioma co-cultures for these tumoroids and organoids. And so we've started playing on with what happens when you have one copy of NF1 missing, two copies of NF1 missing, and adding in 9p loss. And so really, really early days. I think as you know, by asking the question, these are long experiments. And so the two things that I can tell you is NF1 minus glioma astrocyte co-cultures actually don't like to grow. So something about being NF1 homozygous actually slows them down. The old data would suggest this is because of something called RAS-induced senescence. They have too much RAS activation, and that tells the cell to senesce. And that's why we think you need CDKN2AB loss to escape that senescence. So we're kind of now jumping into putting both in and seeing if we can do that. The other thing I can tell you is that the NF1 plus minus tumoroids or organoids actually behave differently from NF1 wild type. So we think there's a haploinsufficiency effect as well. So these are things that we had speculated but didn't really know. And so in like 5 years, if you invite me back, I could give you the update on it. But that's the amount of time I expect it will take. Yeah.
One of the other differences between your NF wild type and NF mutant gliomas was that I think there was a increased incidence of PTPN11 mutation in the NF mutants.
Yeah.
Are those kind of canonical activating mutations? Do you think that it's a RAS dosage effect? What's going on?
Yes and yes is the short answer. Okay. We didn't annotate them as pathogenic unless they were a canonical activating mutation. So I guess the question, sorry, for everyone else is PTPN11 gain of function mutants, I should clarify, were enriched with NF1 loss, but not NF1 mutant wild type tumors. So PTPN11, I had it on one of the slides. I didn't belabor it because I didn't talk about it today. It's a phosphatase domain containing protein that's required for SOS to activate RAS. So essentially SOS cannot be a GEF if PTPN11 is not there. So because of that, people have looked at PTPN11 as a therapeutic target for a long time because it's really attractive. It blocks every single RTK theoretically from activating RAS. So you kind of hit it at the source. How that plays with NF1 is interesting. So if you think about it, taking a step back, NF1 is a gap. So if you lose NF1, you have more GTP loaded RAS. Do we really need PTPN11 at that point to GTP load more RAS? This data and other people's work suggests yes. So I think there is a dosage effect. Whether that is therapeutically useful is unclear. There are the gene that encodes, the PTPN11 encodes is SHP2. So there are a number of SHP2 inhibitors out there in the world. So I think early days, but there is a trial concept floating around to use a SHP2 inhibitor and NF1 for this reason.
Within the context of GMML and
yeah.
PTPN11,
yeah.
So it feels like a difference.
I also wonder if the the JML and rhabdo are, I think they're mostly deletions in NF1, right? Not point mutants. We have a ton of point mutants. So I also wonder if there's a hypomorphic effect of some NF1 variants where PTPN gives it a boost. It's not statistically significant, but we did think about this and we actually broke them down. Are NF1 like truncating or deletion or nonsense variants enriched in PTPN11 wild type versus mutant? And there's a trend, but I just don't think we're powered to say anything. Awesome. Thank you.